Good morning, students. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, Hyderabad, is here to talk on rhizosphere. The term rhizosphere was first coined by Lawrence Hitler in 1904. He was a German scientist. What is a rhizosphere? The region of the soil in contact with the roots of a plant is known as rhizosphere. It contains many microorganisms and its composition is affected by root activities. In other words, it is an environment under the influence of the plant. The rhizosphere was defined by Lawrence Hitler as the soil compartments influenced by the roots of growing plants. Root zone. A very narrow zone, a few in size around the roots, is known as root zone, contains root exudates and microorganisms. Rhizosphere is relatively stable, nutrient rich environment. The rhizosphere is a zone surrounding the roots of the plants in which complex relations exist among the plant, the soil microorganisms, and the soil itself. The plant roots and the biofilm associated with them can profoundly influence the chemistry of the soil, including pH and the transformation of mineral compounds. Here is a picture showing a rhizosphere surrounding a root of a growing plant. In the picture, we can see the root, different parts of the roots, stellar region, cortex, root hairs, then mucilage, root capsules, and root cap. These are the parts of the plant roots, and around these roots the region the region around this is defined as rhizosphere, and the very first layer on the root is known as rhizoplane, and here we can see the association between the roots and the soil. What is a rhizoplane? Root surface adhering soil particles. Microorganisms intimately associated with root surface are mostly fungi like Mozzarella, Cephalosporium, Trichoderma, Penicillium, Gliocladium, Gliomatrix, Fusarium, Cylindrocarpon, Botrytis, and Coniotrium, Mucar, Pythium, Aspergillus, like this, various types of fungi can be found or isolated from the rhizoplane. Here we can see the picture of the first picture in the Petri plate is Mozzarella, followed by Cephalosporium. There are different pictures of Cephalosporium. And the first picture is Botrytis, second one is Trichoderma. Fusarium, the very first picture is Fusarium, followed by Penicillium and Mucar. Next, moving on to rhizosphere effect. It is a direct influence exerted by the plant roots on the microorganisms within the rhizosphere. Compared to non-rooted bulk soil, the rhizospheric soil around the plant root contains much larger population of microorganisms. A stimulation that can be put on a quantitative basis by the use of R is to S ratio. R is to S ratio is equal to number of microorganisms in the rhizosphere is to number of microorganisms in the non-rhizosphere. When the ratio is greater than 1, it is known as good stimulation. When the ratio is equal to 1, no stimulation, and when it is less than 1, it is inhibition. Plant affects the rhizosphere microorganisms through the release of root exudates. What is root exudate? Small molecules that are released from the plant roots, which include sugars, amino acids, organic acids, and amides. These molecules influence soil nutrient availability both directly and indirectly by stimulating the activities of certain microbial and fungal components of the soil biota. Organic compounds released by plant roots are sugars and polysaccharides, amino acids, organic acids, fatty acids, sterols, growth factors like auxins and gibberellins, enzymes, flavonoids, and nucleotides. The rhizosphere region is highly favorable habitat for the proliferation, activity, and metabolism of numerous microorganisms. The rhizosphere microflora can be enumerated intensively the microscopic, cultural, and biochemical techniques. Microscopic techniques reveal the types of microorganisms present and their physical association with the outer root tissue surface or root hairs. The cultural technique most commonly followed is serial dilution and plate count method, which reveal the quantitative and qualitative population of the microflora. At the same time, a cultural method shows the selective enhancement of certain categories of bacteria. Biochemical techniques used are designed to measure a specific change brought about by the plant or by the microflora. The rhizosphere effect on most commonly found microorganisms via bacteria, actinomycetes, fungi, algae, and protozoa. Microorganisms in the rhizosphere. Firstly, bacteria. The greater rhizosphere effect is observed with bacteria R is to S values ranging from 10 to 20 or more than with actinomycetes and fungi. Gram-negative, rod-shaped, non-sporulating bacteria, which respond to root exudates, are predominantly in the rhizosphere, like Pseudomonas agrobacterium, while gram-positive rods, coca, and aerobic spore-forming bacteria like Bacillus and Clostridium are comparatively rare in the rhizosphere. The most common genera of bacteria are Pseudomonas, Arthobacter, Acrobacterium, Alcaligens, Azotobacter, Mycobacterium, Flavobacter, Cellulomonas, Micrococcus, and others have been reported to be either abundant or sparse in the rhizosphere. From the agronomic point of view, the abundance of nitrogen fixing and phosphate solubilizing bacteria in the rhizosphere assumes a great importance. The aerobic bacteria are relatively less in the rhizosphere because of the reduced oxygen levels due to root respiration. The bacterial population in the rhizosphere is enormous in the range from 10 to the power of 8 to 10 to the power of 9 per gram of rhizosphere soil. They cover about 4 to 10% of the total root area, occurring profusely on the root hair region and rarely in the root tips. There is predominance of amino acids and growth factors required by bacteria are readily provided by the root exudates in the region of the rhizosphere. Next, moving on to the second type of microorganism, fungi. In contrast to their effect on bacteria, plant roots do not alter or enhance the total count of fungi in the rhizosphere. However, rhizosphere effect is selective and significant on specific fungal genera like Fusarium, Verticillium, Aspergillus, Penicillium, which are stimulated. The R is to S ratio of fungal population is believed to be narrow in most of the plants, usually not exceeding to 10. The soil serial dilution and plating techniques used for the enumeration of rhizosphere fungi may often give erratic results as most of the spore forms produce abundant colonies in the culture media, giving a wrong picture. Example, aspergilli and penicillium. They almost occupy the entire, a single colony occupies half or an entire plate, which gives a wrong picture. In fact, the mycelium forms are more dominant in the field. The zoospores are forming lower fungi such as Phytophthora, Pythium, Aphanomyces are strongly attracted to the roots in response to particular chemical compounds excreted by the roots and cause diseases under favorable conditions. Several fungi, example Gibberella and Fusicori, produce phytohormones and influence the plant growth. Next group of microorganisms, Actinomycetes, protozoa, and algae. Stimulation of actinomycetes in the rhizosphere have not been studied in much detail so far. It is generally understood that actinomycetes are less stimulated in the rhizosphere than bacteria. However, when antagonist actinomycetes increase in number, they suppress bacteria. Actinomycetes may also increase in number when antibacterial agents are sprayed on the crop. Among the actinomycetes, phosphate solubilizing like Nocardia streptomyces have a dominant role to play. As rule, actinomycetes, protozoa, and algae are not significantly influenced by their proximity to the plant roots, and their R is to S ratio rarely exceeds 2 to 3 is to 1 and around roots of the plant. R is to S ratio of these microorganisms may go to high. Because of large bacterial community, an increase in the number or activity of protozoa is expected in the rhizosphere. Flagellates and amoebae are dominant and ciliates are rare in the region. Root exudates and their chemical substances. Amino acids, all naturally occurring amino acids are found in the root exudates. Organic acids like acetic, butyric, citric, fumaric, lactic, malic, propionic, succinic acids are found. Carbohydrates like arabinose, fructose, galactose, glucose, maltose, mannose, oligosaccharides, raffinose, ribose, sucrose, xylose, etc. are found in the root exudates. Nucleic acids like adenine, cysteine, guanine, uridine are present. Then growth factors like biotin, choline, inositol, pyridoxines, etc. may be found. Vitamins like thiamine, nicotinic acid, biotin, etc. Enzymes like amylase, invertase, protease, phosphatase are found. Auxins, glutamine, glycosides, hydrocyanuric acid, peptides, UV absorbing compounds, nematodes, attracting factors, spore germinating stimulators, spore inhibitors. Other compounds are also found in the root exudates. Changes in rhizosphere microflora. The changes in rhizosphere microflora are reported to take place by soil amendments, foliar application of nutrients, artificial inoculation of seed or soil with preparations containing live microorganisms like Estobacter, Biseringia, Rhizobium, and phosphate solubilizing microorganisms bring about changes in the natural microflora of the rhizosphere. Thank you.